Okay, are we rolling?
I think we are.
All right. I am Spencer Wells, founder and CEO of Insitome, and I'm here with Razib Khan,
Director of Scientific Content.
And this is our first AMA, Ask Me Anything. Any questions you might have about genetics, human history, archaeology, anthropology, linguistics, paleo this, paleo that, ancestry testing, and of course, Insitome's products. So any questions? Let's see if we can kick it off. I am not seeing the video at the top, so I'm going to rely on somebody to feed me the questions. I'm not seeing any questions. Okay. All right. Okay, first question. Is the out of Africa theory in crisis? Well, so let's step back a little bit and talk about what out of Africa theory is. For a long time, it was widely debated in paleoanthropology circles where humans had originated. Some of the earliest remains in the 19th century were actually found in Asia, down in Java. So a paleoanthropologist named Eugène Dubois found Java man at the end of the 19th century. And so there was a lot of debate, well up into the middle part of the 20th century about where modern humans had originated. And then starting in the 1960s, the Leakey family, of course, started to find all these amazing fossils all over Africa. And it became widely accepted that Africa was the homeland of our species. This was confirmed when we started adding DNA sequence data, genetics into the mix in the 1980s. So mitochondrial Eve in the late 80s, early 90s, Y chromosome Adam, which came a little bit later in the 90s and the early noughts. All of the evidence pointed very clearly to a recent African origin, our species, Homo sapiens, originating in Africa within the last couple of 100,000 years, and a subset of Homo sapiens spreading out of that continent to populate the rest of the world. So that was the nice, simple story until very recently. And of course, it's gotten a little bit more complicated since then.
Yeah. So I guess let me, Spencer's knows this stuff like the back of his hand, obviously, involved in the Y chromosome, as most of you probably know. I generally agree with that. What I would say is, as far as if you dropped into the landscape of like population genetics, paleoanthropology in the mid-2000s, this was an issue where it was not even a theory anymore, like saying the theory of evolution is a theory. People accepted this as a very strong orthodoxy. Within paleoanthropology, a lot of people, it seems, believe that there was an out of Africa event that was a massive expansion 50,000 years ago that was total replacement of all the other human species, human lineages all around the world. Things have changed step by step, incrementally and gradually. There hasn't been a paradigm shift, but there's a lot of uncertainty right now. First of all, in about 2010, you had the DNA that showed that there was Neanderthal admixture into humans outside of Africa. You will get different answers from different disciplines about how probable this was. Many population genetics people will tell you privately and publicly that this should not be entirely surprising. There were papers as early as the late 1990s that showed that there wasn't enough statistical power in the mitochondrial lineages to reject the idea that there was admixture, et cetera, et cetera. But if you talk to paleoanthropology people and human evolution more generally, especially in the popular culture, the 2010 results, Green et al. Out of Svante Pablo's lab about Neanderthal admixture were a huge shock. And I think that was...
Huge shock. I mean, the story, of course, was that we had driven Neanderthals to extinction.
Yep.
And there's a mitochondrial sequence that was published by Matthias Krings and Svante Pablo's group in 1997, where the mtDNA falls well outside the range of variation among humans. And so the story was, even based on the genetics, there's no mixing between modern humans and Neanderthals. And then this result comes along, which is a huge shock.
And there are I've gotten to discussions with human population genetics about this. Some of them say, well, we didn't have enough power, et cetera, et cetera. But I do point out to them that before 2010, there were publications in genetics that showed inferences how it was basically impossible for admixture to have occurred looking at the data. So this is an issue where scientists have certain expectations, and sometimes, frankly, they see what they want to see. Since 2010, there have been other things that have occurred outside of simply genetics. So probably the reason this question is being asked is there was a find in Israel, just I think last week it was published in Science.
Yeah.
Do you want to like talk about paleoanthropology and how that's changed in the last couple of years with the Morocco finds? And,
I mean,
yeah.
So the oldest well-dated modern human remains in the world were found at Omo-Kabish in Ethiopia by Richard Leakey back in the early 1970s. Early dating was about 130,000 years ago. They were redated about 20 years ago, and 195,000 years old. So that was the story: is that you got DNA saying we originated in Africa 200K. Seeing the same thing in the fossil record, and then there's this very well-studied find at Jebel Irhoud in Morocco. Remains that were recently reevaluated, and they look a little bit more modern than people suspected, and so they date to about 335,000 years ago.
Yeah, they have a wide interval, but it's a huge interval.
But well before this 200,000-year-old date, and so suddenly that raises the question. Number one, we're no longer in sub-Saharan Africa. Number two, we're much older than we suspected. So where do you draw the line? What do you start calling Homo sapiens?
Yeah.
What came before? Was it Heidelbergensis? Does it grade very gradually into Homo sapiens? Or is there this sudden appearance of modern human traits, which was the story until recently? The new find in Israel dates to about 170,000 years ago, and it shows that things that looked very much like us, so they were definitely Homo sapiens, were living outside of Africa at that time. 170,000 years ago isn't so much later than Omo-Kabish. And so suddenly the question becomes, is it clear that we originated in Africa, or could it have been more of a back and forth between Eurasia and Africa over the course of hundreds of thousands of years?
Yeah, and it's not just paleoanthropology. The fossils and the genomics are moving forward to add to the uncertainty. So to give a concrete example, 10 years ago, people would say, well, modern humans derive from, and this is like stylized, there's variations across disciplines. Modern humans derive from a tribe that lived in East Africa 50,000 years ago, and that tribe expanded, and that's why we have a population bottleneck. That's a stylized model that was very, very prevalent about 10 years ago. With genomics, researchers over the last couple of years have been able to see that there's a lot of deep structure in modern human lineages, and in particularly within Africa. So to give you a concrete example, it was about 3 years ago, I was talking to a friend of mine who works in human population genomics, and he was saying that in his lab, they just estimated the Khoisan, the San Bushmen of South Africa, they diverged from other human beings 200,000 years ago. This is considerably older than that 50 to 60,000 year date, or even 100,000, right? And with ancient DNA, that has been confirmed or even pushed back from 200,000. And they might not even be the most early diverged lineage of modern humans within Africa. So I don't want to get into the technical details because this isn't established science, it's all provisional. But it looks very, very clear that there was a lot of population genetic structure within Africa before the 50,000 years ago event that we used to call the out of Africa event. So the out of Africa event in classical population genetics, using a few markers, you can see a huge bottleneck effect out in non-African populations. But within Africa, that's not nearly as clear. So it's not that we've moved to a new model and the theory in crisis has been overturned. We're in transition towards a more refined and precise understanding of what happened in the past. And that precise understanding is not parsimonious. Right?
Absolutely. And, listen, the simple story that I told in Journey of Man 15 years ago, this recent emergence from Africa, complete replacement of everything else that might have been out there at the time. We now know that that probably isn't correct, and it was much more complex. And ancient DNA is really kind of helping to lead the way here. A lot of the big discoveries in human population genetics in the last few years have come from the analysis of ancient remains. So to summarize all of this, the story's gotten a lot more complicated in the last few years. But the one thing we do know is that it's not as simple as a direct out of Africa model replacement of everything within the last 50 or 60,000 years.
Yeah, it's not a theory in crisis, it's a theory in evolutionary modification.
Which is fun. It's a really exciting time to be working in the field.
And it's a time that we have to update content all the time.
So let's go on to the next question. This is a little bit lighter one. Razib,
all right.
What is your Y chromosome haplogroup?
Oh, yeah.
I think everybody knows that.
Yeah, I'm definitely R1A, which is well known to be the most superior of all lineages. I mean, the discoverer...
I beg to differ as an R1B.
Yeah, well...
Anyway, tell us about R1A.
So R1A, it's very common in South Asia, parts of Central Asia, and Eastern Europe. So it's all over Eurasia. It looks like about 4,000 years ago, there was a massive expansion of R1A lineages. If you look at the ancient DNA combined with the genomic inference. And it's part of a broader story that we have been arriving at as geneticists and researchers over the last 5 years, that during the Holocene, over the last 10,000 years, there's been an explosive expansion of particular Y chromosomal lineages. Now, Spencer was involved in the early 2000s in the work with the Genghis Khan haplotype. And it seems that this event of the expansion of Genghis Khan or elite Mongol lineages is the latest in a series of such events where paternal lineages have expanded and blown up massively in the past. And R1A is not the only one. R1B is another one.
Yeah, I mean, we think of them as what we call star haplotypes.
Yeah.
And one of the first examples of this that we really detected in the genetic record was Genghis Khan's Y chromosome. Very famous finding that we published in 2003. Came out of a lot of work that my colleagues and I have been doing, many expeditions over the course of a decade to Central Asia, collecting samples all over the place. And we found this lineage that seems to have originated in Mongolia based on diversity and frequency, and been driven to very high frequency throughout the extent of Genghis Khan's former empire. And so the idea is that a lineage that's very young, originated in the last 1,000 years or so, is suddenly found at a frequency of 10, 15, 20, even 50% in some populations. What could have driven that? Well, it was probably culture. It was probably the fact that Genghis Khan was a very powerful guy. He had a lot of wives, did a lot of conquering.
Yep.
Took a lot of concubines. His sons inherited that proclivity, for lack of a better term. And this Y chromosome lineage spread very rapidly. And so 1 in every 200 men walking around on Earth today is a direct descendant of this guy who lived in the 13th century. So it's a pretty amazing result. R1A is another example of that.
Yeah.
R1B to a certain extent. There are a few of these. And they reveal something really interesting about human history and about the tempo and mode. So let's move on. Okay. Can you expand on the complexities of inferring ancestry from genomic data?
Mm-hmm.
Okay.
Do you want me to take that?
Yeah, sure. We just did. So this our latest podcast.
Yeah, our latest podcast with Joel
is all about this.
The Insight. You can find it on iTunes, Stitcher, and through our Twitter feed and Facebook. But we talked with Joe Pickrell and Jen Cove about this and the different choices the different companies make about how to infer the ancestry. So the key issue is, what is the question? First of all, you start out with, what is the question that you are asking? I can create a genetic test that confirms that you're a human being, right? That's true, but it's also trivial. So you want to ask a question that's not trivial, that gives you novel insight, some sort of surprise, right? And the surprise needs to be robust. It needs to be genetically valid in terms of the framework of a model with data. So when I'm going into designing an algorithm or a framework to infer ancestry, I want to make inferences that are intelligible or comprehensible to the public if it's a direct-to-consumer. If it's a scientific publication, that's a whole different thing. It's a whole different. So for example, like give a concrete, in ancient DNA, David Reich's lab and few other ancient DNA labs, they use relatively abstruse names for ancient population cluster, like early European farmer. Now, some of the people that, frankly, probably most of the people that are watching this live feed, they will probably know what that means. But when you're talking about a general public audience, they don't know what early European farmer means. And so you have to work with units, population units that are comprehensible to them. They know what Southern European means in general because it's a geographic term, so you're going to use a lot of geographic terms. And in terms of the data, we have a lot of data now. That's not really a major issue. So when Spencer started, when he did Journey of Man, for example, for autosomal data, which is anything besides the Y and the mtDNA.
There was virtually nothing.
Yeah, X. There was not that. There was hundreds of markers in Cavalli-Fortz's lab and a few other places.
But that's why the Y chromosome and mitochondrial DNA were state-of-the-art in those days.
Yeah.
Because they don't recombine, you could start to build up these sets of linked markers, which allowed you to construct a tree, a stable phylogenetic tree, start to talk about haplogroups. because you could date things using mutation rate. You could talk about the ages of the lineages. And so those uniparental markers, inherited only down the male or female line, it gave us really the first glimpse of how we moved out of Africa to populate the world. Based on autosomal evidence, prior to that time, the only thing we could say is that there was more diversity in Africa than outside, and so it was consistent with an African origin. But in terms of the journeys around the world and the routes that we took and the timing of settlement of these various regions, that really came down to the line of the mtDNA.
Yeah, and in terms of inferences and using genomic data, another thing is there's been a lot of discussion among scientists now that, oh, well, these are misleading, and it's not telling us real history, and it's not where your ancestors are from. I think that's a fair objection, but the reality is if you're going to give ancestry results to regular people, they are going to make historical inferences. So my goal when making decisions about which clusters to analyze or which clusters to pull out of the data is to maximize historical inference possibilities for the consumer, And so, basically the questions are actually more important in some ways than the method or the data, because we have a lot of data. And the method...
The questions help you to kind of mold the analysis
Yes.
to provide the answers that people really want.
Yeah.
And, unfortunately, I think a lot of people, the reason these tests are so popular in places like the US is that we're a nation of immigrants and everybody is a hyphenated American. And so most Americans know more about the stuff that comes after the hyphen, i.e. the American part, than the stuff that comes before. So if you're Irish American or Italian American, it's a very vague sense of, your connection to those places. And so that's what people tend to be looking for.
Yes.
The problem is countries are very recent inventions
Yep.
for the most part.
Nation states,
And,
yes.
Italy, I always use this as an example, fewer than, half of the Italians actually spoke the Italian language at the time Garibaldi united Italy in the 19th century. It was a bunch of kingdoms and duchies.
I think it was like 10%.
Exactly.
It was a Florentine dialect.
And so it's got these groups that were actually quite different from each other that are suddenly thrown into a single country. So if you're defining people as Italian, that actually encompasses a huge range of variation. So it's hard to detect that genetically.
Yes.
And that's just one example. Same thing for Germany, same thing for Britain and other parts of Europe.
Yeah, and with our audience, it's probably on this live, they understand that you wouldn't want to say that there's like an Argentinian genetic ancestry, but I watch a lot of YouTubes about Ancestry Reveals, and this woman was wondering, her great-grandfather was from Argentina. She's Lebanese. She was wondering, oh, does she have Argentinian blood? That's not a thing. Argentinians are mixed people just like Americans. They're settler people. They're not American genes, unless you want to count Native American, right? So the way we frame it is really important, and we try to do a good job, I think, here. And that's our goal. We want people to have like true results that they can understand, right? And that's not always trivial. That's not always trivial.
Okay, what? Okay, so this touches on a similar point. So this is someone who's writing in asking about, okay, I've tested my Y chromosome, my mitochondrial DNA. They tell me about these uniparental lines. So your Y tells you about your father's 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 father. Mitochondrial DNA tells you about your mother's 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 mother. And if you think about that growing tree of ancestors where you double in every generation, then you go back a few generations and you're missing the vast majority of people who contributed to your DNA. The person asks, well, what about those other people? And that's really these autosomal tests.
Yeah.
And that's the reason they've really come to the fore as kind of the go-to for understanding your ancestry, because they encompass all of your ancestors.
Sure. Well, let's think about it. Let's give a concrete example of how this could differ. So, I was reading some paper about Argentina, admixture, et cetera, et cetera. This is a, most Argentines identify as European white, but if you look at their whole genome, they're about 20% native, give or take. If you look at their mitochondrial DNA, their maternal lineages, they're about 50% native. 50% look native. If you look at their Y chromosomes, it's about 90% European, something like that, more than 90%. What is this telling you? It's telling you that Argentina was subject to migration of men from Europe, mostly Southern Europe, over the last couple of hundred years, right? And so if you have mitochondrial DNA that shows your direct maternal lineage, your Y chromosome that shows your direct paternal lineage, they're going to be telling you very different things. If you look at your autosomal ancestry, you see that it's somewhere in the middle in terms of like these different ancestral pools that have different sex ratios, et cetera. So you need all three to really understand your genealogy.
Okay, we have a question that has come in. What is the future of regional ancestry mapping? Will the resolution ever get down to regions within a country? And will Y-DNA and mitochondrial DNA be used for a product at Insitome? So several questions. Let's start with the first couple of parts. What's the future of regional ancestry mapping? How precise do you think we can get?
You know, this is a matter of our reference samples, and I mean, mostly it's the reference samples, and like perhaps some changes and improvements in the method. It depends on the country. So to give you a concrete example, China is a big country, and there is a non-trivial, there's a big distinction between North and South Chinese. So obviously, I can see us splitting that apart within the year.
And when you say reference samples, I mean, what we're talking about here, the way we do this is we make use of data that's been collected from so-called indigenous populations that have been living in the same place for a long time.
Yeah.
We try to collect as many samples as possible from all over the world, and to drill down into subregions within a country or within, say, Northern Europe or Central Europe, you've got to have a lot of separate population samples from people that have been living there for many generations in basically the same villages. And so it's not easy to get the samples. I mean, I ran the Genographic project for over a decade, and that's what we focused on, was working with indigenous people to get that database and build it up. In some cases, we do have the data. So I think Britain is a great example. It's been very well studied now, the people of the British Isles Project, or POBI as it's called.
Yeah.
And there is actually a company called Living DNA that makes use of this
Yeah.
to really kind of drill down on your ancestry within Great Britain. And
Yeah.
I actually think it's a really good product because I happen to have a lot of British ancestry.
Yep.
You're not British, not necessarily so
Nope.
useful, but I think if we could do this around the world, and we're making an effort outside of Europe. So in India, for instance, we've made choices about the reference population so that we can really start to drill into variation that you see within the Indian subcontinent.
Yeah, there's a lot of low-hanging fruit outside of Europe, and we're definitely going to go for that fruit. We're going to keep going towards Europe. There are certain countries where drilling down is going to be a problem because we don't have the reference populations, and the countries are also very homogenous, And then there's other countries where it would be pretty easy, actually. China was one example. I do think in knowing what I know about Italy might actually be a good candidate within Europe for doing a North or South Italian cluster.
Oh, definitely, because you're going to have a signal. In fact, we see this. I mean, this was one of the studies we did, Genographic. Just looking at Sicily, you can see the layers of migration that have come in from North Africa, from the Middle East, the Norman invasion, and obviously a connection to the Italian mainland. And then you can see very distinct differences between northern and southern Italy that has to do with the population history.
Yeah.
I mean, part of the complexity in conveying ancestry results to people is that we, as students of history and prehistory, we kind of understand the archaeology and the linguistic patterns. And so when we see something like a north-south difference in Italy, it makes complete sense to us because we get the history behind that. But if you're expecting all Italians to be the same, it's a little confusing. And so it's really a question of us. The onus is on us as the company that offers the results to explain that population history to the extent that we can.
So I want to actually call out to a question that came in from Americo Ross, great name by the way. How much European DNA could have been spread into North Africa and the Levant by the Roman Empire, and how is it taken into consideration?
It's a good question.
Right? So again, the terms you use matter a lot. There are some European populations in Southern Europe that are as close to some West Asian populations or North African populations as they are to Scandinavian populations, right? So how you draw the boundaries matter a lot. The difference between Europe and non-Europe was not as stark during the Roman Empire. The Mediterranean was the Roman Sea, and cold Northern Europe was the outgroup, right? But right now, if you had to put me on the spot, it doesn't look like there was that much migration that has survived down to the present. It might be because people mostly moved amongst, the mobile people moved among cities, which are population sinks. There's less perturbation in the Mediterranean than history would lead us to expect, okay?
Well, so just to back up a little bit, and the Romans weren't the only ones moving around.
Yeah, Greeks.
So, and the Phoenicians. So I was involved in the
Yeah.
long-term study with my colleague Pierre Zalua at Lebanese American University, and we sampled all over the Mediterranean, North Africa, Lebanon itself. And the culmination of all of this was a paper that we published several years ago, where basically we showed that 1 in every 17 men in the Mediterranean basin is likely a descendant of the Phoenicians.
The white Renaissance.
But a lot of that is localized around these population centers. So Carthage was, effectively the Phoenician capital, after about 700 BC. So it was no longer an Eastern Mediterranean collection of peoples. It was a North African collection of peoples. They controlled the tin trade, made them fabulously wealthy. And of course, the Romans came in, very jealous of the fact that they were wealthy traders, and they destroyed them in the Punic Wars. In the Third Punic War, literally, they reduced Carthage to rubble the size of golf balls or smaller. And it's very sobering to walk around the remains of that once great city. So I think there were impacts, but we're not talking about more than 5% or so.
Yeah, when I've looked into it, the main area where I think that there might have been a lot of things that happen that we can detect in terms of population churn is maybe Sicily. And Sicily was under Arab Tunisian rule also for 2 centuries. You can see evidence of this as well. So the Sicilian, and they're in the middle of the Mediterranean. They're on the border between the west and the east. So it's difficult. What I would say is, to a first approximation, there has been a surprising level of genetic continuity in the Mediterranean from antiquity to the present. So there's been some ancient genomes from Lebanon, from the Bronze Age, about like 1800 BC. They look similar to modern Lebanese. Now, I have looked at enough Christian minorities or just like non-Muslim groups in the Middle East versus Muslims. The Muslims tend to be more cosmopolitan. They have some East Asian, probably from Turks. They have some African, probably from Sub-Saharans. Although in general they're very similar to the Christians, they just have other things going on. So it's more like the Roman Empire was an inflection, kind of a different flavor on top of what was the base that was already there. It didn't change the base, from what I can tell, to a first approximation. There are places where that's not true, but those are the exception of the rule.
Well, let's talk a little bit about ancient DNA. We keep mentioning this. And, I recently, we released a new edition of Journey of Man last year. And we're
going to do a podcast on Journey.
We're going to do a podcast, JOM Redux, we're working on it now.
Yeah.
But, they asked me to write a new foreword. And one of the things I talked about in the new foreword is the advances that have happened since that book was published in 2003, so in the last 15 years. And the 2 big advances in my mind are, the simplicity or the cheapness of whole genome sequencing, especially with the dawn of next-gen sequencing in 2007 or so. You can now sequence a whole genome for $1,000-ish. If it's low coverage, it's a tiny fraction of that. But also, ancient DNA. That's a huge advance.
Yep.
And, I often tell people, asked many times when I give public lectures or something, like, if you could change anything about a geographic project today, what would it be, knowing what you know now? I'd probably spend more than half the money these days on ancient DNA.
Yeah.
Because that's really providing us this glimpse into the past. It's a time machine. Yeah. I mean, you're literally traveling back in time.
Yeah.
You've got well-dated archaeological layers, carbon dated, you know exactly what culture they're related to or part of. And you can literally go in and take a sample and discover changes in the DNA as different cultures move through. It's fascinating.
Yeah. So let me this ancient DNA thing. If you study phylogenetics, and phylogenetics doesn't actually have to deal with just genes, you deal with linguistics. In any case,
so this is the relationship between populations.
Evolutionary trees.
Yeah.
If you study phylogenetics, the way you do it in genetics, just the way you do it in general, is you have this model of trees. Just divergence, evolutionary divergence, and you're sampling from the tips of the tree. And by sampling from the tips of the tree and seeing the patterns of the tips of the tree, you can find the way the branching occurred within the tree. That's what really we're talking about here, concretely. What ancient DNA did is it gave us samples from the internal parts of the branch, right? And so all of a sudden, giving samples on the internal parts of the branch, not just what we infer, what we kind of guesstimate, but the reality of what it was, brought a lot of aspects of the tree into really, really sharp resolution, because just with the tips of the tree, you cannot necessarily differentiate well different hypotheses sometimes. Different admixture scenarios can give the same genetic pattern. But if you have the internal branches of the tree, the nodes as we call them, all of a sudden you get much more clarity, much more precision. And
you see things that nobody ever saw before. I mean, the best example is obviously the Neanderthal genome.
Yeah.
That gives us an insight into mixing between Neanderthals and modern humans. But I think what's even more startling to me is somebody with, say, British ancestry, is what some of the Bronze Age remains.
Yes, and we actually have a question related to that. I'll read it off real quick from Russell Burgess. I had hoped that the last 2 books by Brian Sykes, Saxons, Vikings, and Celts, and the book on the genetic roots of Britain and Ireland would shed light on our origins. As you study detailed DNA from the British Isles, however, nowhere in the books were haplotypes or markers discussed. Are you aware of where this information can be obtained? So I read the first book about over 10 years ago now. It's seriously out of date. In terms of information on British genetics is the most well-studied human genetic question in the whole world at this point. Partly it's historical because genetics to some extent modern genetics comes out of Britain. So there's a 2015 paper out of the POBI, Peopling of the British Isles, Bodmer's group's paper. It's in Nature, look for it, Peopling of the British Isles, you can Google it. That will tell you the structure of the modern British, right? There's also a preprint, Bell Beaker, go to bioarchive, Bell Beaker, and put David Reich, put R-E-I-C-H. That's ancient DNA. And that shows the ancient stuff.
And the story is startling.
The story is
frightening.
So here, yeah, it is a little frightening. So if you guys have ever watched the beginning of Conan the Barbarian, there is a scene where the soldiers of Thal Doom show up and they slaughter the whole village except for Conan. And by the way, the Sumerians, Conan's people, are supposedly the mythological ancestors of the Gaels. That's all made up, but that's just kind of coincidental. I joke that that's what happened in Britain 4,000 years ago because the ancient DNA shows us that before about 2,500 BC, the people, the farmers in Britain, and these are the people that succeeded the hunter-gatherers who were genetically totally different. So there's already one transition. The farmers, they resembled modern-day Sardinians genetically, right? So these were the Neolithic farmers that brought agriculture, and they were the ones
from the Middle East.
Yes, and they were the ones that actually set up Stonehenge, by the way. Stonehenge dates to 3000. So they were megalith builders, right? So these were the first megalith builders. What happens around between 2500 and 2000 BC, over those 500 years, probably a narrower date, but let's be conservative or give a wide interval there. About 80 to 90% of the genes turn over in terms of there was almost total replacement.
I.e., the people died.
Yeah.
It's not like they had anywhere to go. They didn't set up in robots for the shop.
We don't know if they were killed. They could have decided to become celibate.
But it is what we call a population replacement.
Yeah,
and so you've got a 90% genetic turnover in the span of a few hundred years.
Yeah.
That
happened that happened in the United States too. 200 or 300 years ago. So I'm just saying human history, the past and the present are not as different as you would think.
But the point is, we wouldn't know about that without the ancient DNA.
Yeah, and so Britain has experienced 2 population turnovers of relatively wide scale since the end of the last ice age. Brian Sykes' original work was predicated on the idea that 80% of modern British ancestor dates from the Pleistocene. That's false. It's probably well less than 10%, perhaps less than 5%, and only a minority of modern British ancestor even dates from agricultural farmers from the Middle East. Most of it actually comes from Central Asian, descendant of Central Asian people that settled in Central Europe and brought the Bell Beaker culture about 4,000 years ago. That is the answer to that question. And read those papers.
Okay. Somebody's asking about testing every region of the world. Every ethnicity on Earth deserves funding to have their genetic history studied and archived. Is there any sort of initiative toward achieving this? Well, this was really the goal of the Genographic Project.
HGDP originally too, right?
HGDP, which never really got off the ground in a major way.
Yeah.
There were maybe 1,000 samples that, quite frankly, existed at the outset that they ended up making cell lines out of. And it's a great resource. But Genographic, we ended up collecting roughly 72,000 samples from over 1,000 populations around the world.
Nice.
And they're stored at the centers, so you can go on the Genographic site or look it up on Wikipedia and see where the centers are based. That's where those indigenous samples are stored. So we had 15,000 from India, for instance. I think it's probably the best sampling of Indian diversity that exists in the world.
Yeah.
So this is an ongoing process, and but at the moment, I don't think there is a concerted effort. There's certainly big studies, largely in European and East Asian countries.
Yeah, those countries are not
to sequence genomes.
Yeah, those countries are not problematic. Let's cut to the chase. Having a concerted worldwide effort is going to run into objections, and people are not going to expend their time.
Yeah, this was a significant issue in the Genographic Project.
Yeah.
I mean, there were objections from indigenous populations who, were concerned that we might be telling them something that they didn't want to hear about their ancestry, or they were concerned simply about what we might be doing with the DNA. There were lots of issues, and so I spent a lot of my time really being a politician during my 10 years head of the Genographic Project. trying to do as much science as I could. It's a non-trivial undertaking, and I feel like we are most of the way there. I mean, I think I would be very surprised to see a result from an isolated population that completely turned everything on its head.
At this point.
I mean, maybe the Sentinelese or something.
Yeah, that's the only one. I mean, most of them, like even 10 years ago or 5 years ago, there are still groups that you would want more sequences from. But at this point, I think we have most of the tree filled in, and there shouldn't be too many surprises. But
the key is to get those ancient samples
from
to
get
places
the
like
samples.
the Indian subcontinent, East Asia.
Yeah, and the ancient samples are also a problem, though, because, if they're indigenous people, then you have to get consent from their putative or potential descendants, but you don't know if they're descendants until you sequence it. It just gets really complicated. Frankly, genetics is easier to do with Europeans and East Asians for political reasons, and also, like disease, like these are the populations that are paying for the research, et cetera, et cetera. It's unfortunate, but that's how it is, right?
Yep. Okay, we've got a question about an Insitome product. So let's change gears slightly. In addition to what's already supported in Insitome traits, so that's regional ancestry, Neanderthal ancestry, and then the ancestral story behind your metabolism, so why you metabolize certain foods in certain ways. What other consumer-facing traits do you plan to focus on integrating into your app in 2018? So good question. I mean, listen, all of these apps, or insights as we call them, because there's a single app that you can download on your phone and then run like individual insights. They're like chapters in your story. So, we've started off with some of the basic stuff. A lot of people are interested in the Neolithic transition and their own metabolism and so on. How does it affect the foods I should be eating in my diet and so on. Neanderthal story, I think, is a really sexy one. Regional ancestry, is probably our most popular app because it just answers that basic question, where did I come from? But all of these are tied together through this focus on your ancestry and how it has an impact on things that affect you. And so, we're going to be telling stories about additional traits, and we're going to group them together thematically, but traits that have a selective or an evolutionary story behind them. And so, for instance, we were just talking about Bronze Age replacement. We've got an example of that we've actually been doing basic research on, and we'll be publishing a scientific paper in the next few months that will end up as one of the traits that you're going to be able to test for. It's actually an adaptation to that bloodshed, if you will, during the Bronze Age. So, all of the traits in this initial app are going to be tied together by a focus on ancestry, but it's really how has your ancestry had an impact on you? It's not just ancestry for ancestry's sake, although, that's very interesting. But it's also like, how does your ancestry affect traits that you can see when you look in the mirror or when you sit down to eat a meal? And so that's really going to be the focus for the coming year.
The bigger contextual framework.
Yeah. And as you like to say, it's phylogeny and function.
Yeah. So we're going to focus on function. We're going to focus on phylogeny. Regional ancestry is all phylogeny.
Yeah.
There's no function.
And we probably will be adding Y and MT DNA at some point.
Yeah.
I suspect. I mean, it's pretty easy to do that. The fact is, though, again, unless you're really interested in tracing your surname with the Y chromosome, or you really have some particular interest in mitochondrial DNA, they're not as useful on their own as genome-wide regional ancestry would be.
Yeah. Let me just a quick question related to Insitome results from Amelia Leveni. She was just asking about why the Geno showed Mediterranean and Geno 2.0, and we did not show Mediterranean. I just want to reiterate, different companies might have the same label for different reference populations. Right? So our North Mediterranean cluster is Central Italian. That's the easiest way to think about it. Geno 2.0, their Mediterranean, I think, is a little bit more cosmopolitan, and so it would have a wider range in terms of who gets matched up with it, right? So I mean, that's the short answer. Similar names, but they're actually different reference populations. So you have to keep that in mind.
Okay. Any other questions coming up? Okay. Well, I think we're probably going to wrap it up there.
All right.
You have anything to add? What are you excited about looking forward in this year, scientifically?
I think there's going to be a lot more ancient DNA. I know there's going to be a lot more ancient DNA. As we talked about on our podcast, we had on India, which we actually recorded last summer, so a while ago. There is a paper that's in the works that will be a blockbuster paper. It might be two papers. To make the long story short, there's a lot of politics involved that's causing issues. There's a lot of rumors about it. But as we've been saying, sometimes people don't get results from genetics that they want, in which case they will slow walk it, or they don't want to confront it. So politics is an issue, especially when you have international collaborations. But so that paper is coming out. I know there's a lot of stuff in behavioral and cognitive genomics that will be coming out. That'll be interesting.
David Reich has a book coming out.
Yes, David Reich has a book. So what's it called? Like how we got here, or who we are? Yeah.
Like
It's coming out in March.
Yeah.
David Reich has a book that's coming out in March. Get it? People have been asking me for years, what book should I read to understand all this stuff? That is the only one that I can name, partly because it changes so fast because of his lab. Steven Pinker has a book coming out at the end of February called Enlightenment Now. That's not genetics
It's got a lot
related.
of press already.
Yeah,
he was
but
at the WEF.
Yeah, but I'm super excited about that because hopefully it'll make me a little more optimistic about the world. But
That would be good.
Yeah. So I'm excited about that.
Anyway, so it's going to be an exciting year, both
Yeah.
here at Insitome and more broadly, scientifically in this field. All right. Thank you very much.
All right, we'll sign off there.